In this set of notes today, we're going to be talking about the wonderful process of photosynthesis. So let's go over the outline for today's notes. I'll start with just a general overview of photosynthesis and its equation, then a brief description of the anatomy of the chloroplast. Then we'll break down the stages of photosynthesis between the light-dependent reaction and the Calvin cycle, and then I will go over an entire diagram review of the process of photosynthesis. So for these notes, you can pause at each slide and fill in the guided notes found in the description below, or you can watch the video straight through. All right, so let's begin. It's important to remember that all life on Earth requires energy to move, to breathe, to grow, to reproduce. All of these actions require energy. So we need an energy source, which for Earth is the sun. So sun is our ultimate source of energy. Plants are going to use this solar energy that we get from the sun and do a process called photosynthesis. So photo means light, and that's going to be our solar part of photosynthesis. Synthesis means to make. So we're going to be making something from this sunlight. So we can go over what is being made by looking at the chemical equation. So since photosynthesis is using solar energy and chemical substances to produce other chemical substances, we can write it out in a chemical equation. The idea is we're taking solar energy and converting it into a chemical form of energy. And that's because living organisms need it in this form. We can't directly use it as solar energy. We need to use energy in the form of a chemical. So the equation is written here below. We start with 6CO2, which is carbon dioxide, and water, H2O. Now, these are the chemical substances. They are made of elements. We also need, the important part here, the solar energy. But I put that going over the arrow as opposed to putting it before the arrow because it serves as one of the ingredients for photosynthesis, but it isn't a chemical reactant like the other two. So once we mix our chemical reactants and solar energy, we're going to produce glucose. That's what we have there, C6H12O6. And this is a form of sugar. So this is a chemical form of energy that we can then use. If you'd like to know what we do with this glucose, you can watch my other video on cellular respiration of how we're going to use that chemical form of energy. And then of course, the last product here is going to be oxygen, those 6 O2s we see at the end. So this is the overall equation of what's going in and coming out of the process of photosynthesis. Now, photosynthesis is unique to plants and some photosynthetic bacterias or protists. Now, animals can't do this and fungi can't do this, and that's because our cells lack the organelle or cell structure necessary to do it, and that is called the chloroplasts. Photosynthesis takes place in chloroplasts, and plants have a lot of these to be able to do this process. So to better understand the stages of photosynthesis, it's good to have a general understanding of the anatomy of the chloroplasts. So this green structure here is a cartoon version, simplified, of what a chloroplast looks like. So inside the chloroplast, we have these green pancake-looking like things. These are membrane enclosed sacs, and they are called thylakoids. So each individual one is called a thylakoid. And you can see that there are a lot of these. And that is because this is the part of a chloroplast where the sunlight will actually get absorbed. So these little thylakoids, our pancakes here, are going to be taking in and absorbing the solar rays as they come in. Around the thylakoids, we have this liquid environment, and we call that the stroma. So the stroma is going to be where the second stage takes place. So our sunlight gets absorbed here, but that actual final product of glucose, that sugar we talked about in the equation, is made here in the stroma. So all this is enclosed in its own membrane here, and this is going to be our whole site of all photosynthesis. Photosynthesis happens in two major stages. So let's start with the first one. The first one is called the light-dependent reaction. Now, all of photosynthesis depends on light, but this is going to be the one where we're actually utilizing the sunlight. So that's where it gets its name from. So if you remember, as I just went over in the previous slide, we have these thylakoids that are membrane-bound sacs. In the thylakoid membrane, we're going to find a series of proteins, and this is where the light-dependent reaction is going to take place. So we're going to start with our first reactant here, water. So water is going to be in our thylakoid, and it's going to be split. Since water is made up of hydrogens and oxygens, the oxygen will break off and the hydrogens will break off when we split this. Once we do that, we're actually going to take an electron from hydrogen and send it into photosystem 2. So photosystem 2 is the first protein that the electron's going to go through in this electron transport chain. The reason it's called photosystem 2 was because it was the second one to be discovered, but it is the first one in the system here. So in this photosystem 2 protein, there's going to be a pigment that allows the sunlight to be absorbed. Different wavelengths of the sunlight will be absorbed. And it's going to do something called exciting that electron. Basically, we're going to energize that electron with the solar energy. And so you can see that's going to push our electron up here. It's energized and excited. Then the electron is going to travel through some other membrane proteins that we have in this thylakoid membrane. As it's going through these proteins, its energy is going to be used to transport more hydrogen ions into the thylakoid, creating what we call a concentration gradient, because we're getting the hydrogen from these waters and we're pushing more hydrogens in, and we're building up a high concentration of hydrogen ions inside the thylakoid, as opposed to outside where there should be a lower concentration. This is going to create that concentration gradient that we need to power a protein called ATP synthase. So ATP synthase is another protein, and its job is to make a molecule called ATP. This is that chemical form of energy that living organisms need. We can't use sunlight directly. We can't even use sugars directly. We can use ATP directly for energy. And this protein, ATP synthase, is designed exactly for what it says, to produce and make these ATP. But it runs on these hydrogen ions. Just like your car runs on gasoline, you're going to need your fuel for this ATP synthase. So since we brought in more hydrogen ions and built up this concentration gradient, we have the hydrogen ions to flow through ATP synthase and produce ATP. Now that electron that we got earlier from hydrogen has been traveling, and it's going to go to the second protein where it's going to get excited, called photosystem 1. So photosystem 1 is the second protein that it encounters as opposed to the first, but they're named for order of discovery, not order in the lineup here. So once this electron that has used its energy to move hydrogen ions across gets to photosystem 1, there's pigment in photosystem 1 that is also absorbing other wavelengths of sunlight, and it's going to re-excite this electron and energize it. So this now newly excited electron is going to be picked up by an electron carrier called NADP+. So this is going to be our taxi driver for this electron. Its job is to take it from one location to another. Once it picks up two excited electrons, it's going to become NADPH and add this hydrogen to sort of cap it off. So at the end of the light-dependent reaction, we have some ATP that has been produced and some NADPH that is carrying some excited electrons. Now we are done with the light-dependent reaction. So we're going to move into stage 2. Now, everything we just talked about was happening in the membranes of these thylakoids here. Now we're going to move outside of the thylakoid into this liquid environment called the stroma. In this stroma, we're going to use our other reactant. So we were using our water and splitting it for those hydrogens and oxygens in stage 1. In stage 2, our other reactant, carbon dioxide, becomes very important. So carbon dioxide is going to enter into what we call the Calvin cycle. And so we're going to break down the cycle step by step, and then I'll show you the whole cycle on one slide. So the first step is carbon dioxide is going to enter the cycle and combine with a 5-carbon molecule called RuBP. And you can see this here. I've made the carbon dioxide blue, the blue carbon with 2 purple oxygens, and it's going to attach to the 5-carbon RuBP. After this connection happens, this bonding takes place, it's going to immediately split into 2 3-carbon molecules. These are called PGA. So we have one 3-PGA that has the carbon we got from carbon dioxide, and we have a second 3-PGA, which is the remaining carbons from RuBP. Now, that ATP and the electron carrier, NADPH, are going to donate these electrons and a phosphorus from the ATP and overall convert 3-PGA to a molecule called G3P. When we do these models and we show pictures, we tend to focus on the carbons. The carbons are really the essential building block that we follow for making our product. But there are hydrogens and oxygens involved, and there's going to be certain bonds that break off and reshape, which is why these molecules are changing names. So in this case here, it doesn't look very different. We went from 3 carbons, we added phosphorus and electrons, did some chemical conversions, but it still looks the same here because we're focusing on the carbons. But this does now have some electrons that it didn't have before and has changed in form on a bigger scale. So now we have these 3 G3Ps. One of them is going to exit the cycle at this point, and it is going to be used to make glucose, that sugar that's the final product of photosynthesis. The other one, the one that was just the 3 carbons left over from RuBP, is actually going to get recycled. That's why it's a cycle. This is going to go back to the beginning and get chemically converted back to a 5-carbon RuBP so that we can restart the cycle. Now, as we went through the steps, I showed you with a single carbon dioxide. But one turn of the cycle actually requires 3 carbon dioxides that are all going through these steps simultaneously. So since glucose is a 6-carbon sugar and we have 3 carbon dioxides in one turn of the cycle, it takes 2 cycles to produce 1 glucose. So if we are starting from the beginning with the amount of reactants that we showed in the equation there, it's going to take 2 turns of this cycle to make the 1 glucose molecule that we need. So I'm going to go over it in a diagram here. We can see we start with the carbon dioxide. That carbon will attach to the 5-carbon molecule RuBP. Then we're going to split it into 3 PGA, 2 of them. The ATP and NADPH that we got from the light-dependent reaction are going to use these electrons and phosphorus to convert it to GP3. And these are now going to be recycled as well. ATP becomes ADP since it lost a phosphate. NADPH becomes NADP+. These will go back to the beginning of the beginning of photosynthesis to continue the process. Then one of these is going to go in to make glucose, and the other one will be recycled. Some more ATP will be used, and we will have the RUDP that we need to continue the cycle. And just a reminder, 3 carbon dioxides are going through this process at one time. So in photosynthesis, we have the overall reaction taking place inside the chloroplast. Inside the thylakoid membrane here, we're going to take in our solar energy and our water to get ATP and NADPH. Those oxygens I talked about earlier that got split off from the water, that's going to make our oxygen molecules that get released and is going to give us that oxygen product that living organisms need for cellular respiration. Then the products ATP and NADPH will leave the thylakoid membrane and go into the stroma, where they will enter the Calvin cycle. And in the Calvin cycle, we're going to take that carbon dioxide and modify it using the energy that we received in light-dependent reaction and use that to produce glucose. So here we can see an overview of our reactants: water, carbon dioxide, and solar energy, and how we get to our products: glucose and oxygen. And then we will use this in another chemical process called cellular respiration. So I hope this helps you better understand photosynthesis and the process of how we get chemical energy from solar energy. If you have any questions, please leave them in the comments, or if you'd like me to elaborate on anything further. Thank you, and I hope this helped you with your biology studies.